Diabetes Insipidus by Shelby Fisher and
Kristine Clara.
The normal anatomy of the nephron. The nephrons are the structural and functional units of the kidney. There are over 1 million nephrons in each kidney. The nephron carries out the processes that form the urine. There are also thousands of collecting ducts that collect fluid from the nephron and transport it to the renal pelvis. Each nephron has a renal corpuscle and a renal tubule. The renal corpuscles are found in the renal cortex, and the renal tubules start in the cortex and go into the medulla. The renal corpuscles have capillaries called glomerulus. The glomerulus have many pores, which helps large amount of fluid to pass from the blood into the glomerular capsule. The filtrate is the material that the renal tubules process to make urine. The glomerular capsule has an external layer which is called the parietal layer. This is made out of simple squamous epithelium. The second layer is a visceral layer which sticks to the glomerular capillaries. This layer has highly modified branching epithelial cells called podocytes. The podocytes stick to the basement membrane of the glomerulus. The filtration slits allow the fluid to enter the capsule space inside the glomerular capillaries. The renal tubule has three major parts: the proximal convoluted tubule, the nephron loop, and the distal convoluted tubule. ADH is produced in the hypothalamus and secreted and stored by the posterior pituitary glands. The function of ADH is to cause the body to retain water and constrict blood vessels.
So what is diabetes insipidus? Diabetes insipidus is a water homeostasis disorder characterized by the partial or total inability of the kidneys to produce concentrated urine due to kidneys' insensitivity to ADH or decrease in synthesis or production of ADH.
The two types of diabetes insipidus are central diabetes insipidus and nephrogenic diabetes insipidus. In central diabetes insipidus, there is damage to the pituitary glands or the hypothalamus. This damage disrupts normal production, storage, and release of ADH. Nephrogenic diabetes insipidus is caused when there is a defect in the kidney's tubules. This is the structure that causes water excretion and reabsorption. The risk factors for central diabetes insipidus are surgery, a brain tumor, an illness like meningitis, inflammation, head injury, and some cases are unknown. The risk factors for nephrogenic diabetes insipidus are genetics, a chronic kidney disorder, or drugs like lithium or an antiviral medication.
Pathophysiology: what went wrong in homeostasis? To maintain water balance in our body, water intake and output should be regulated. This regulation involves the sensing of plasma osmolality by the osmoreceptor. Osmoreceptor is a specialized nerve receptor found in anterior hypothalamus which have projections to TERS and vasopressin center in the hypothalamus. Normally, when there is a change in blood osmolality, osmoreceptors in hypothalamus is stimulated to produce ADH and delivered it to the posterior pituitary gland. Posterior pituitary gland then releases ADH to the bloodstream, causing vasoconstriction of the blood vessels, and ADH also goes to the nephrons to the collecting duct to inhibit aquaporine insertions for water reabsorption back to the bloodstream to bring back the blood osmolality in the homeostatic level. In diabetes insipidus, it's either the posterior pituitary gland that doesn't make enough ADH or the nephron is unresponsive to ADH, causing uncontrolled blood osmolality, which causes excessive production of hypotonic urine. So what went wrong in the cell membrane? In the collecting duct, by channel-mediated facilitated diffusion, the controlled water reabsorption happens. ADH or arginine vasopressin now bind with AVP2 receptor with a chain reaction. It activates G protein, then activate the enzyme adenylate cyclase. Adenylate cyclase uses ATP and it converted into cyclic AMP. Cyclic AMP activate inactive protein kinase which activates aquaporine storage vehicles to be attached to the cell membrane, allowing water transport back to surrounding capillaries. When there is lack of ADH production or ADH receptors doesn't respond to ADH, water reabsorption is uncontrolled by osmosis, which results to excessive urination of diluted and hypotonic urine, then followed with excessive thirst. Signs and symptoms are excessive urination because of uncontrolled water reabsorption and excessive thirst because of excessive urination.
Now for the summary. Central diabetes insipidus is caused when there is damage to the pituitary gland or hypothalamus. Nephrogenic diabetes insipidus is caused when there is a defect in the kidney's tubules. This disturbs the process of excreting and reabsorbing water. Homeostasis is disrupted because there is not enough ADH production or the receptor does not respond to the ADH. In the cell membrane, water reabsorption is uncontrolled by osmosis because of the lack of ADH. Proteins allow water transport back to surrounding capillaries. Without facilitated diffusion with the use of aquaporin channels, AVP2 receptors, which are proteins, reabsorption is unable to happen.